So in this video, I'm going to talk about the discovery of genes and materials of heredity. So first, let's talk about genes. So what is a gene? Well, genes are located on chromosomes. So essentially, it's a segment of a chromosome. And remember, chromosomes consist of DNA and protein, the histones. And so at first, people thought that both could possibly be genetic material. And in fact, proteins, since they have more variability, they were thought to be associated with heredity. So people thought that histones were actually associated with heredity. But, however, we know that that is wrong. And so let's talk about how people discovered that. So the first person that we're going to talk about is Griffith. And so what he did was he looked at pneumonia in mice. And so he essentially, there were two strains of bacteria, R and S. S, in red, killed the mice, while R, the R strain, did not. So first he injected S into the mice died, and then he injected R into the mice, and the mice did not die. However, here is where it gets interesting. So what he did was he heated the S, and we'll just use this as a mice, that face. And so he heated the S and killed the bacteria and injected it into the mice. And so what happened was the mice then did not die. So now what he tried was he heated the S, but then he killed the S and combined it with the R. So dead S plus R, injected it into the mouse, and then that resulted in the mouse dying. And so now he was trying to figure out what caused that. And so he took the blood from these mice and discovered that they had live S bacteria. And so he was just wondering what happened, like what caused that. And so that process is called transformation. And that's essentially when the S virus, the virulence of S was passed to the R. And it converts R to S. And it converted R to S. And so that was what Griffith discovered. Now, let's talk about another experiment, Hershey and Chase. And so what did they do? Well, they used viruses. And remember, viruses are not technically considered alive because they cannot reproduce on their own. They need a host, and they are specific to a host cell. And so they use viruses, more specifically bacteriophages. Which essentially are viruses that infect bacteria. And so bacteria has a protein coat and a nucleic acid core. Nucleic acid. And so the key difference between protein and nucleic acids slash DNA or RNA is that the nucleic acids have phosphorus. And so DNA was essentially labeled with radioactive phosphorus so it could be detected. And so they labeled the DNA in the viruses with radioactive phosphorus in order to detect it. And the viruses infected the bacteriophage, so virus infected the bacteriophage. We'll use red for the labeled phosphorus. And then what happened was it turned out that the bacteria was they detected radioactivity in the bacteria. Then they tried this for protein to use radioactive sulfur, because protein has sulfur and nucleic acid does not. And they found out that the viruses were radioactive. And so this allowed them to conclude that DNA was the genetic material. So DNA was genetic material.